Good morning, children. In the last class, we were discussing about in the topic post-fertilization structures and events. We have discussed until the formation of the endosperm and embryo, right? Today we will continue with respect to the formation of seed from ovules. Okay. See, observe the structure. Here there are two diagrams which is given, the dicot seed and the monocot seed. If you see the structure, you can identify there are two cotyledons in the dicot seed. What about in the monocot seed? Yes, it containing single cotyledon. Yeah, let us start the discussion. So with respect to the meaning of the seed, seed is the fertilized ovule formed inside the fruits. It is nothing but the product of the post-fertilization, isn't it? Yeah. It is the final product of the sexual reproduction, and also it consisting of the seed coats. There are two, the coats which covers the seed, outer testa, inner tegma. And in case of the dicot, two cotyledons are present. What about in case of the monocot? Yes, a single cotyledon is present along with the embryo axis, so that you can identify clearly the embryonal axis also, isn't it? Yes. The cotyledons are simple. Generally, they are thick and swollen due to a storage food. So generally, the cotyledons are clearly identified and observed in case of the legumes. So that in general we can also say the pulses what we used in our homes as a food products, isn't it? So just observe the structure. I hope you can identify the seed coats. The seed coats, its outer testa and inner tegma, and the hypocotyle, epicotyle, and the cotyledons. The endosperm is a nutritive tissue. Yes, in case of the monocots also you can identify. Yeah, got it? Then, and observe, see, these seeds are generally, so they are classified into two types. So based on the presence or absence of the albumin. So albumin is also known as endosperm. So generally the albumin, the terminology used in the zoology, so but here also it is understood that. So there are two types of the seeds. The classification. When it comes to the classification, we can identify the mature seeds are of two types. Just observe here it's also. So based on the presence or absence of the albumin, as I said, if they have no residual endosperm, as it is completely consumed during the embryo development, so such a type of the seeds are called non-albuminous. Non means absence. No residual endosperm. That means nutritive tissue is absent. Okay. Why it is absent? So during the developmental stages it is completely consumed or utilized. Okay. So such a type of the seeds are known as non-albuminous. We can give number of examples. So that is the pulses are the best examples, like pea, groundnut and beans, which can be given example for the non-albuminous. In case of the albuminous, they retain. That means, yes, the albumin or the endosperm is present. Even though after the developmental stages, so they retain some endosperm. A part of the endosperm is retained. That means it is present. Such a type of the seeds are known as albuminous seeds. Example, so maximum in case of the wheat, maize, barley, and also castor, coconut, and sunflower. While we are consuming the maize, you can identify a milky like appearance, and in case of the castor also, it is in the form of the oil, right? Yes, the castor oil. And the coconut, while we are chewing the coconut also, we will identify, that means milky like appearance, isn't it? And also we will extract the oil from the sunflower seeds, right? Yeah, so these are all nothing but endosperm. So that's why they are also used as a food products as well as in the form of the oils also. Okay. So they also provide nourishment. So castor oil is used as the hair oils. Sunflower oil is used as a eating oil. Got it? Yeah. Here are the diagrams they have given, the model structure and also the paizoms sativum. That means commonly known as pea or Gordon pea. Yeah, that is the stored form of the cotyledons are the larger part, outer testa, inner tegma, the seed coats, the plumule, then hypocotyl, radicle and embryonal axis you can identify. So when it comes to the monocotyledon, it is an example. Maize seed actually, and also we can take example of the corn also. Yeah. So here it is in case of the monocot. That's what I said. Whenever we chew, we will identify a milky-like appearance, right? Because of that is endosperm. It containing large amount of endosperm, right? So that's why the milky-like appearance you can identify. Observe neatly. Yeah. Okay. So here are the textbook diagrams also. These diagrams which is present in our textbook. Here also you can identify the cotyledons, the micropyle end, opposite to that one chalazion, and the seed coats. You can identify two layers of the seed coat. That's what I said. Outer testa, inner tegma. Then endosperm, the cotyledon, and also you can identify embryonal axis. Are you getting? Yes. So here in case of the structure of the monocot seed, generally they will ask sometimes in exams also. Last year also you have studied, right? Outermost region or the layer is nothing but peri, means above, outermost layer, the pericarp. Then the endosperm. Are you observing? Then plumule is well protected by coleoptile, and the radicle and the root system is protected by coleorhiza. Are you getting? Yes. So then after this one, you can identify some more examples. Yeah. So here it is some of the rare cases they have given. The perisperm. Occasionally, generally these type of the cases are rare, but we generally observe. In some seeds like black pepper, beet, you must have observed after opening up this black peppers, we can identify the whitish nature, right? Yeah. So that is nothing but the remnant of the nucleus. Yes. In some seeds like black pepper and the beet, the remnants, that means which is containing. It is not completely utilized. Actually, nucleus is a nutritive tissue before the fertilization, which is present in the ovules, right? Yes. But that will be not completely utilized. It will be retained. A part of the nucleus is retained after the development also. So then that structure is nothing but perisperm. The remnants of the nucleus are also persistent. Or we can also say the persistent of nucleus is called perisperm. Got it? Yeah. Then the integuments. If you observe clearly, here it is given, there is another example, pepper seed and also the pea seed, that is peanut seed. The botanical name is Arachis hypoxia. Yeah. The integuments of the ovules harden as tough protective seed coats. It has a small pore. That the pore region is nothing but micropyle, through which oxygen and water enters into the seed during germination. Are you getting? Yes, the integuments is generally a protective layers, the outer integument and inner integuments. Well, we are studying the structure of the in the beginning only. The anatropous ovule we have studied, right? Outer integument and inner integuments. They are nothing but protective seed coats. But the micropyle end, which provide the That is nothing but the entry of oxygen and water into the seed during germination. Okay, yeah. So here you can identify the how the germination of the seeds will happens. As the seed matures, it becomes a dry by reducing water content. It's about 10 to 15% moisture by mass. So then what will happen? That temporarily the metabolic activities of the embryo slows down. It may enter a stage of inactivity, or we can also say a state of inactivity is called dormancy. The reason is whenever the pulses are the seeds, that means 10 to 15% of the moisture, it will reduces. Immediately there will be name of the hormone which is called dormin, which is present. It is get activates, so that it may enter the stage of the inactivity. That is nothing but dormancy. Okay, then after that, if we provide the proper, the moisture, oxygen and suitable temperature, it is also known as seed treatment, right? Yeah. Under favorable conditions, it may be provided by artificially or naturally. So whenever under favorable conditions it is provided, or they make it naturally, they they become active, they start germinate. Got it? Yeah. So these are all the stages, developmental stages of the seeds. Then this get converted into seedlings. Are you observing the stages of development of the seeds? Yeah. Then the question arises, is there any usage? Yes. The advantages of the seeds, not only for the human seeds, it is also for they have advantages towards the plants also during the period of the evolution for the survival. Since the pollination and the fertilization are independent of water, the seed formation is more dependable. Are you observing? The pollination and the fertilization are independent of water. That means there is no necessity. Pollination and fertilization are independent of water. Even though in the absence of water also it will takes place, the seed formation is more dependable. Okay, yeah. The better adaptive strategies for the disposal to the new habitats. While we are studying the earlier classes, we know about the dispersal of the seeds, right? Yeah. So they may dispose about kilometers together. Then what will happen? For the better adaptive strategies, they have to survive. If there will be no better adaptive strategies, they will not survive, isn't it? Easily seeds will be get destroyed. So that for that purpose, the better adaptive strategies for the disposal to new habitat, the seeds formations helps the species to colonize in other areas, so that after the disposal also they will survive, they will grow, and they will furnish in new environment and in new habitats also. Yes, third one, they have food reserves. So young seedlings are nourished until they are capable of photosynthesis. Yeah. So whenever the seeds, that means matures, what will happen? Until they undergo a cyclic changes and they become independent, until they become capable to photosynthesis independently, they need a nourishment, right? So that is provided by, that is food reserves. As we have discussed, it is a cotyledons. Got it? Yeah. And even in case of the endosperms, so then even mucilage, they have food reserves, right? So these food reserves, so young seedlings are nourished until they are capable of photosynthesis. After that, so that the green leaves, they will start photosynthesis, then they can lay independently, isn't it? Yes. And also the advantages, the hard seed coat protects the young embryo. So I told you number of times. So whenever the seeds disposes, It may be influenced by biotic and abiotic agents. Sometimes due to the water and air it may be disposed. Sometimes due to the animals, isn't it? In such a cases, the hard sheet coats outer testa, inner tegma. It protects the young embryo. Got it? Then, and being product of sexual reproduction, they generate new genetic combination. That is nothing but degree of differences. There is nothing but variations. Okay, during sexual reproduction, what will happen? The male gametes and female gametes fuses together. They give rise to the new generation, so that the variations is possible. Then there is another advantage. Dehydration and dormancy of mature seeds are crucial for the storage of seeds. It can be used as food throughout the year and also to raise crops in the next season. That's what I said. That the dehydration, whenever the percentage of the water will reduces 10 to 15%, they undergo dormancy. Because of this feature or the characteristic feature, we can store the pulses also for sometimes about years together. Got it? Yes. And so here are some of the examples. The ability to survive, isn't it? That means the ability of the seeds after the disposal. There are a number of examples. Until then, it is recorded. Here are some of the diagrams. Observe this. In a few species, the seeds lose viability within a few months. That means ability to survive. Okay. Then, the seeds of many species live for several years. So that is depends and differs with respect to different species. Here are some examples they have given. Some seeds can remain alive for hundreds of years. One of the oldest one recorded, the oldest is that of Lipinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record. It's about, you cannot believe, it's about 10,000 years of dormancy. After that also, yes, it is activated and it is grown. That was the estimated record. And there is another example. 2,000 years of old viable seed is of the date palm. The botanical name Phoenix dactylifera. It is discovered during the archaeological excavation at the King Herod's Palace near the Dead Sea. So these are all some of the classic examples. Okay, so how the seeds they will protected years together. Okay, yeah. And after this one, the last, the topic in the post-fertilization structures, the fruits from ovary, right? Here are some of the examples they have given. Observe the structure. Yeah. We observing. Yes. The fruit generally will be considered as, that is a ripened form, isn't it? Before that, generally we will not consider it's a fruit. Generally the ovary develops into a fruit. The ripened form generally we will identify as a fruit, isn't it? But actually here it is also, it will be containing, yes, the seeds inside the fruits. It will be containing the fruits from the ovary. The fruit is also arises from the ovary only. It is majorly divided into three regions, that is epicarp, okay, then mesocarp and endo. Epicarp is also known as exo, meso means middle region, endocarp, okay, and inside that one seeds will be present. Peri, so this, what are the structure which covers the seed? These structures will be considered as a peri, means above. Okay, if I take an example of the mango, you can identify these structures clearly: exocarp, mesocarp and endocarp. Mango, outermost region, the peel, it will be considered as exocarp, and the meso, the edible fleshy mango, and endo, which is covered, the hard coat which is surrounded the seed. Inside that one, the seeds will be present. Okay. The transformation of ovules into seeds and ovary into fruits proceeds simultaneously. Okay. So this will be takes place. That means simultaneously at a time. The wall of the ovary develops into the pericarp wall of the fruit. That's what I said. Then the fruits may be classified into two types of fleshy fruits and dry fruits. We can give number of examples: guava, orange, mango, citrus fruits. Almost all they are all considered as a fleshy. If you take example of the dry, that means groundnuts, mustards, they are also considered as a fruits only, technically. But in generally we will consider as a seeds, of course, but technically and botanically they are also nothing but the fruits only. Okay. And here are some of the examples, the fruits from the ovary generally. So again the classification of fruits based on the development which is from the ovary or not. Okay. The fruits are majorly classified into two types, that is true fruits and false fruits. What is the meaning of the true fruits? If the fruit is developed from only from the ovary, then they are nothing but the true fruits. Okay. And the remaining other floral pods degenerate. For example, if you take from the ovary only, remaining layers and additional floral pods, it will degenerate. Okay. So then such a type of the fruits are known as true fruits. False fruits, sometimes other than the ovary. Okay. For example, here there are given example in case of the apple, strawberry, and cashew. These fruits are arises from other than ovary. Actually, that is nothing but thalamus. Observe the diagram. The apple, what we edible, the edible structure is the white color, right? Yeah. That edible structure of the apple is nothing but thalamus. Even in case of the strawberry also. Okay. While you are eating, you must have experienced strawberry may containing the minute and numerous number of seeds. But the fleshy region, what we consume, that is nothing but thalamus. In case of cashew also, you may think that the cashew is nothing but dry, but actually it is arises from the extra, that is the thalamus region. It will be extended outside the fruit. Okay. So these are nothing but false fruits. Okay. What is a false fruit? If the fruit is not arised from the ovary, that means in case of these apples, strawberry and cashew, thalamus which contributes the fruit formation. So such a type of the fruits are known as false fruits. Yeah. And there are some cases, the fruits from the ovary. Okay. So and not from the ovary. And there are even some specific cases. If you would have observed, you must have, generally, a number of cases, you must have heard the word, seedless. How it will be. In some species, fruit develops without fertilization. Such fruits are called parthenocarpic. Banana is actually natural parthenocarpic. If you would have observed neatly, banana is not containing any seeds, isn't it? Yeah. But how it will be gives rise to the plants? Yeah, rhizome of the banana. Okay. So it will be gives rise to the new plants, isn't it? Yeah. Then the banana is the best example for natural parthenocarpic, that is parthenocarpic fruit. What is parthenocarpy? That is nothing but the development of the fruit without fertilization is known as parthenocarpy. Okay. So artificially induced parthenocarpic fruits are nothing but generally seedless fruits. You can give number of examples. Seedless grapes, isn't it? Nowadays, recently they have developed seedless pineapple also, and even seedless. They are developed recently. These are all nothing but recently. Okay, in watermelon also they have developed actually. Yeah. There is a last topic, the apomixis and polyembryony. Apomixis is the production of seeds without fertilization. Okay, such seeds are known as apomictic or apomixic seeds. Okay, so best example asterisk. So here on the left side we have given the number of examples. Marigold, even sunflower, these are also comes under asteraceae only, and even the grasses. Okay, so apomixis is the production of seeds without fertilization. During the reproductive evolution, it is nothing but for the purpose of survival. Okay, so it is a form of asexual reproduction and mimics the sexual reproduction also. So here are some of the examples. So these are all nothing but apomictic seeds. The phenomena is known as apomixis. And the last one is polyembryony. Yeah, before that polyembryony, let me to explain briefly this development of the apomictic seeds. In some species, what will happen? The diploid egg cells is formed without reproduction division. That means what happens generally whenever they undergo, during sexual reproduction, what will happen? They should be get divided. The diploid is get converted to haploid, right? But there are some specific species, as I said now only. Yeah, what are those species? Yes, so asteraceae and grasses. Some species diploid egg cell is formed without reduction division and develops into the embryo without fertilization. So that is the phenomena behind the apomictic seeds. In many species, example like citrus and mango varieties, some nucellar cells surrounding the embryo site divide, protrude into the embryo sac to form the embryos. Thus, each ovule contains many embryos. Okay, many means number is more. So this phenomena is known as polyembryony. Occurrence of more than one embryo in a seed is called polyembryony. The best example you must have observed, if you cut the citrus, lemon only in our homes, we can identify easily number of embryos, isn't it? Mango you should observe clearly, then only you can identify, or else you cannot identify. Okay, they are nothing but the nucellar cells surrounding the embryo sac divide protrude into the embryo sac to form the embryos. Okay, so such occurrence of the more than one embryo in a seed is called polyembryony. Yeah, so here are some of the importance of the apomixis in hybrid seed industry nowadays. So this is the the test tubes which is given in the tissue culture lab. We can identify if the seeds from the hybrids are sown, the plants in the progeny will segregate and lose the hybrid characters. Hybrids, you know very well that by crossing two different varieties for the purpose of the whatever we require, the desirable characters, we will conduct the hybrids, right? Yeah. Then this production of hybrid seeds is costly. So hybrid seeds are also expensive because for the purpose of the desirable characters only, they must have done the number of experiments repeatedly and they have developed in the labs, right? Yeah. If the hybrids are made into the apomictics, there is no segregation of the characters in hybrid progeny, so farmers can keep on using hybrid seeds to raise new crops. If they, no segregation is, they can retain the same character. If there will be segregation, what will happen again? The desired characters will not arise in the next generation. Got it? Yeah. So that's why nowadays hybrid seeds are very costly. You must have, the farmers and children may know about the value of the hybrid seeds. Why they are very costly? because they have conducted the various experiments by trial and error method in the laboratory for the purpose of the particular, the characters only. They have conducted number of trials. So during this process, so they have produced. That's why this production of hybrid seeds is costly. So hybrid seeds are also expensive. Got it? I hope so all of you understood this class. So if you have still any doubts, let me talk clear in the next coming classes. Thank you. Have a nice day.